In cardiac surgery involving cardiopulmonary bypass, it's associated with the risk of acquired coagulopathy, which can be complicated with life-threatening bleeding. So this involves hyperfibrinolysis, meaning the excessive conversion of fibrin into fibrin degradation products, leading to accelerated clot breakdown and bleeding. And aprotinin, an antifibrinolytic agent, has been recommended for the prevention of these complications. Clinicians may at present choose from 3 commercially available antifibrinolytic agents. So we have tranexamic acid, epsilon aminocaproic acid, and the third one is of course aprotinin, which in Europe is currently only indicated for prophylactic use to reduce blood loss and blood transfusion in adult patients who are at high risk of major blood loss undergoing isolated cardiopulmonary bypass graft surgery. And after careful consideration of the benefits and risk, as well as the consideration that alternative treatments are available. This restricted indication is mainly due to findings from 3 observational studies and 1 randomized controlled trial suggesting increased mortality in patients treated with aprotinin. Nevertheless, many clinicians currently use aprotinin outside of its approved indication because it appears that aprotinin is more effective than the other 2 in reducing blood loss and allogeneic blood transfusion, and because recent observational research suggests that aprotinin does not have an inferior safety profile. Its effectiveness has been attributed to its ability to non-specifically inhibit various serine proteases involved in the coagulation and fibrinolysis cascade, such as thrombin, plasmin, kallikrein. Additionally, aprotinin may protect platelets from cardiopulmonary bypass-induced dysfunction through a platelet-sparing effect, further enhancing its efficacy. With this scoping review, we aim to investigate this platelet-sparing effect through the existing literature, as well as synthesize the biochemical effects of aprotinin on blood platelets and to summarize the clinical relevance of its platelet-sparing effect. We included systematic reviews, experimental studies, and observational studies involving healthy humans, patients, or animals undergoing any type of surgery, or studies which involved donated blood for in vitro research if they of course investigated aprotinin and a potential platelet-sparing effect. 3 databases were used for our review, Medline, Cochrane, and Embase, and we ended up with 64 studies in which we looked for a platelet-sparing effect, if this effect is dose-dependent, and if this effect had any influence on blood loss and allogeneic blood transfusions. Of the included studies, 82% observed a platelet-sparing effect, attributing it to one or various of the following mechanisms. So, inhibition of platelet aggregation through protection of the glycoprotein 2B3A receptors, or due to inhibition of platelet adhesion through protection of glycoprotein 1B receptor. Both could be inhibited as well. We also saw inhibition of the proteolysis of the protease activated receptor 1 receptors, as well as inhibition of platelet activation through inhibition of plasmin or inhibition of platelet activation through thrombin. The dose dependency of the platelet-sparing effect was investigated in both the in vitro and in vivo studies, with 62% of the studies showing an increase in platelet-sparing effect when the aprotinin doses were increased as well. However, no studies have explored the relative contribution of aprotinin's platelet-sparing effect and its antifibrinolytic effect in reducing bleeding and need for transfusion. So we wanted to research the full mode of action of aprotinin as its platelet-sparing effect could benefit patients undergoing cardiac surgery who are also on antiplatelet therapy. So the Nordic Aprotinin Patient Registry collected data from 83 European centers to monitor its safety and found that 1,766 patients out of the 5,309 to be under active antiplatelet therapy. So further studies are definitely wanted to investigate whether aprotinin's platelet-sparing effect offers significant clinical benefits in this particular patient population. And another topic of considerable clinical interest is how aprotinin modulates platelet endothelial interactions, which has not been thoroughly addressed yet in the literature. So aprotinin may have a significant impact on these interactions by reducing platelet activation, stabilizing the endothelial cells, and maintaining a balance between coagulation and fibrinolysis. This combination of effects could potentially reduce dysregulated platelet adhesion and aggregation triggered by cardiopulmonary bypass and or inflammation. More research can elucidate the precise mechanisms of aprotinin on platelet endothelial interactions and address the existing gaps in understanding these interactions across various clinical contexts.